the impacts of SDG on the biogeochemistry of corals, going from nutrients, calcification, and mostly carbon dioxide, how SDG can be a source or maybe a sink of CO2. So I'm going to start with the definition of SDG. There is lots of discussion in our groundwater community about what exactly SDG means. Many people perceive groundwater as something fresh, something that we can drink, a resource. Indeed, many of those global water budgets we can find in textbooks, they highlight only the fresh groundwater components. In this illustration, for example, you can see that groundwater discharge to the ocean is only about 10% of the river fluxes to the ocean, but the uncertainties are much larger. So we have maybe a 70% uncertainty for fresh groundwater flows to the ocean, and maybe 10% on the rivers. That's the perception from a hydrologist perspective. I have a marine science background, and my definition is broader than that. So we think that SDG is the flow of water on the coast, continental margins, and that includes both fresh groundwater, the conventional water, and the seawater that penetrates the sediments. It stays in the sediments for some time and comes back out with a different chemical composition. So you can see those processes on the diagram, number 1, 3, and 4. There are things driven by waves, tides, density inversions that push the seawater into the sediments, and that seawater comes out usually very hot in CO2 and many of the tracers, geogenic tracers like radon and radium isotopes that we use. So with this broader definition, it brings questions. The 3 words, submarine, groundwater, and discharge, they essentially mean where, what, and how. So the challenge of the definition is that the word submarine implies something under the sea. But in our definition, it's the coast that includes the intertidal zone, including in coral reefs. The word groundwater, sometimes we think that's something fresh, drinkable, but in the SDG definition is actually brackish, can be actually 100% seawater that goes in and out. And the word discharge can be even more confusing because discharge implies one-way flow, like in rivers, from the continents to the ocean. But in our definition here, it's actually can be a two-way process in that the seawater goes in and out. So it's the same seawater, but with a very different chemical composition. So we use this definition that's broader because all this water coming from the sediments is going to give us a biogeochemical signal. So from a biogeochemical perspective, both fresh water and seawater that goes in and out, they give us nutrients, carbon, greenhouse gases. So that's an example from a coral reef. Right here, I have a photo from Heron Islands in the Great Barrier Reef. And we have the physical evidence for tidal pumping, which is one of the many ways that SDG can be visualized. So in this case here, the coral reef lagoon, during high tide, goes up on the beach, penetrates the beach, and drains back to the ocean at low tides. And you can see those little channels. They are little erosion channels from the seepage of the seawater from the previous high tides. And when that's going to the ocean, it has a very different chemical composition. So this is SDG to me. So the thing about coral reefs, I love this old diagram. This paper from the 80s by Hatcher, really highlighting that corals, they really live in this nutrient-poor ecosystem in that they recycle everything. So the recycling mechanism is highly efficient. So the nutrients that go there, they're taken really quick and they stay in the system. So when you visualize this process, it's essentially a recycling mechanism. The beach is functioning like a sponge that holds the seawater for a few hours and recycles the nutrients and returns that to the coral rather than sending to the open ocean. So during that recycling process in beaches, we're having the release of things like nutrients. That's a summary of all the data available for all coastal ecosystems, not only coral reefs. In that, there was a comparison between the fluxes from rivers and groundwater. So the diagram on the bottom is showing the number of study cases. And here we have the ratio of the nutrient fluxes between groundwater and rivers. Any study or location that plots on this side of the diagram, we have bigger fluxes from rivers than groundwater. And anything on the other side, groundwater is more important than rivers in that specific location. So what we find from this type of analysis is that in about half of the cases where the two things are compared, the groundwater fluxes of nitrogen, phosphorus, and silica, they can be greater than the river fluxes. Of course, there is a bias in that type of assessments because when you do those studies, maybe you are targeting locations where we know or we expect groundwater to be important. But regardless of that, if you go to a random location, maybe on half of the occasions, this broad definition of SDG is going to give us as many nutrients as rivers. If you go to coral reefs, in some cases you have extremely high concentrations of nutrients in groundwater. That's a conceptual model from Heron Island, where we have a very small sedimentary island with carbonate sands. This island has about 10 birds per square meter. It's an amazing amount of birds that bring the nutrients from the ocean. They bring nitrogen from the ocean. They spend the night resting on the trees. And the guano from those birds penetrates the aquifer that exchanges with the ocean, with that tidal pumping process. So the concentrations of nitrates in that groundwater, they can be even higher than sewage. They're very, very rich, more than 1,000 micromolar, millimole concentrations. Right? Of course, as that nitrate reaches the ocean, you're going to have a consumption. The corals and anything that's alive is going to use up that nitrate as quick as possible. So you have a very clear shift in the type of nitrogen from nitrates in groundwater to organic nitrogen in the coral reef. So in that concept of the beggar's buckets, we have all the nitrate being used up really, really quickly, and then that returns to the islands with the birds in this case. So that case, we have a very clear relationship between nitrates in the coral reef lagoon and radon. And radon is my groundwater tracer in this case. That's what's telling us this water was in contact with the island sediments. So the correlation is strong enough. And we know that the nutrients, because there is so little in those places, those nutrients are going to have an impact on the ecosystem production and the calcification. So there is lots of scatter in that relationship, and that only holds true during the day. At night, those black circles here, the correlation collapses because calcification, photosynthesis, they need light. So the nutrients can only stimulate production during the day in those corals. So just to introduce you a little bit about radon, which is our key tracer. So when you make measurements of radon, we're essentially saying that that seawater that we're measuring was in contact with the sediments, and that fits our broad definition of SDG. So radon is natural. It's very high in groundwater because it comes from any geological materials. Radon is a gas. So as a noble gas, there is zero interactions with biology. So biology is never going to interfere with our radon concentrations in seawater. But as a gas, radon wants to escape to the atmosphere. So that makes the concentrations in surface water very low. They're negligible unless you're very close to that source, which we know it's pretty much only groundwater. It's also radioactive. Another reason for radon to disappear. So we say that radon has a short memory. If the groundwater penetrates the ocean, it's going to give us radon only for a few hours or maybe a few days, because it's a gas and as a radioactive element that's going to disappear. And the beauty is that we can have automated systems to make observations of radon, in real time. We can deploy those instruments in the ocean and get back later to collect data. So that has been the basis for the research we're doing on coral reefs. So that's how those deployments may look like. Can be from a small boat, or can be from a box that you deploy carbon dioxide instruments, radon. We have batteries for power. We're pumping the water to an exchanger system that G-gases, that picks up the gases from the water, and then you can go home with time series of radon and CO2. And that has been the main tool to study those systems. So to highlight that it's going to work, I'm going to start by talking about a very new paper that came out a couple months ago. That's one of our PhD students, Apra Tomer, and she compiled the radon CO2 datasets from all these coastal ecosystems, and that includes data from 8 coral reefs. You can see one on the Cook Islands that was part of Tyler Cironak's work as a PhD student. We have a few in the Great Barrier Reef. We have one off the coast of Australia in Lord Howe Islands, and we have the Philippines, for example. So the time series that we collect, they look like that. That's an example from a coral reef. So we have, in this case, 90 hours of data, which, 3-4 days of data. We have a tidal cycle, so the tides are going up and down. In this case, about 1 meter tidal cycle. Temperature is fluctuating. And we also have diel cycles. So oxygen that you see in green here is following the expected diel cycles. So we have the highest oxygen concentrations during the day following photosynthesis. No surprise here. And we also see the CO2 values that have near the opposite trends as oxygen. So photosynthesis and respiration are driving opposite trends for CO2 and oxygen. It thinks that CO2 has a secondary signal that you cannot see in the oxygen. You can see those ups and downs that are related to tidal cycles that actually match the radon peaks. So radon in blue, we have the occasional peak, and they happen at low tides, not following diel cycles. So we have 2 drivers, overlapping drivers, photosynthesis and respiration for CO2, and we also have the groundwater that's high in CO2. If you kind of go to the entire data set, each one of those dots is an ecosystem. And they're trying to separate the diel and the tidal effects of into CO2, radon, and oxygen. So a tidal effect of 0 means that the tides play no role. And if you go here for like one of those ecosystems, that would be like a mangrove. The coral reefs are in pink, so those are coral reefs. So the tidal effect on CO2 for most coral reefs tends to be kind of proportional to the diel effect. So you have just combined tidal and diel effects. When you look at radon, that's our groundwater tracer, most ecosystems, including coral reefs, they plot on just tidal dominated. So tides, they drive radon because groundwater is a tidal process in pretty much all of those ecosystems. When you go for oxygen, the final graph, so we have more of a diel effect. Occasionally, we have a tidal effect. It's quite variable. So CO2 is responding to biological processes and also to the hydrological process. We can see that from this analysis. Now, if you plot CO2 versus our 2 proxies, a hydrology proxy, radon, and oxygen, for all the ecosystems, you can see this cluster of data. Our corals are in pink on the bottom here. So CO2 never gets so high in coral reefs because of the high production. But there is a correlation between CO2 and radon in coral reefs that tends to be positive. The correlation between CO2 and oxygen tends to be negative in coral reefs. And the correlation to salinity is quite variable. So that's telling us that occasionally you have some fresh groundwater that can be a source of CO2. Occasionally primary production can be the most important driver. And sometimes you have groundwater as the key driver of CO2 in corals. On average, you have SDG with stronger correlation coefficients in 5 ecosystems, all the ecosystems with tides, and biological production tends to be more important in coral reefs and the coastal lakes. But groundwater is still there as a secondary driver. So the thing is that groundwater is highly enriched in CO2. So the graph is showing the ratios between groundwater and surface water PCO2. So anything above 1 will tell us that the groundwater is high in CO2 and therefore a potential source. So in some cases, the groundwater PCO2 can be more than 100 times greater than the surface water. So a little drop of groundwater would be enough to raise the CO2 in surface water. In coral reefs, that can go anywhere from maybe 60, 70 times greater, which are looking at numbers like of several thousand parts per million, and sometimes comparable or just a little higher than surface waters. So highly variable in coral reefs, but also highly variable in all these other ecosystems. Okay, so we also try to use radon to build mass balance models and quantify the fluxes of SGG-derived CO2. And here we are showing the fluxes for all these types of ecosystems and highlighting corals. So what we see for corals is that the groundwater flux would be in the order of 74 millimole per square meter per day. And those numbers are much greater than the flux of CO2 at the water-air interface. So there is more CO2 coming from the groundwater than CO2 escaping to the atmosphere in nearly all the ecosystems, and that's more pronounced in coral reefs. And the reason for that is that some of that CO2 that comes from groundwater is going to be taken up by the carbonate system, some of that will be taken up by biology, and some of that is just going to travel offshore and escape to the atmosphere further offshore. But you can see that there is a local impact of SGG in CO2 in coral reefs, and that has implications from the perspective of simple carbon budgets or maybe even ocean acidification. Now we also have these other greenhouse gases. The methane and nitrous oxide trends, you don't have as many data sets, but we do have for this coral reef, Heron Island, one of our favorite places for this type of research. So here we have maybe 10 days of data or so, and you can see the CO2 trends that have this double impact of diel and tidal cycles, and we can also see the methane and the nitrous oxides having these ups and downs, these dramatic changes over just a few hours in this coral reef. So it's easier to visualize if you just plot those three greenhouse gases versus, again, our two proxies, radon, a groundwater proxy, and oxygen, a biology, a primary production proxy. So the data here was broken down into nighttime in black and daytime kind of just blue. So what we see is that CO2 is going to correlate to radon pretty much only during the nights. So the groundwater input doesn't have anything to really counteract that CO2 enrichment at night. But during the day, the CO2 is going to come in, and that's going to interact with biology much quicker. Methane has a strong correlation to oxygen, to radon, both during the day and at night. The correlations to oxygen are much weaker. So there's no surprise that methane is coming from groundwater. We need to have conditions with no oxygen to produce methane. And now nitrous oxide has another unique behavior in that the correlations are much stronger during the day than during the night. So now the nitrous oxide is essentially coming from the nitrogen from the birds. So those birds are giving away nitrates, and that's producing the greenhouse gas nitrous oxides during the nitrification. And during the day, some of that nitrous oxides can actually be used up by biology. When they run out of nitrates, they can turn nitrous oxides to produce biomass. So you can see a groundwater impact on the three greenhouse gases here, but each one behave in a different way. So that was our direct evidence. Now let's look at the more complex processes happening in coral reefs. So the thing is that when you have SGG, it's not only a source of CO2, it's a source of nutrients that drive productivity. And the productivity is going to drive POM, biomass, that's going to penetrate the aquifer and be mineralized there and eventually return to the ocean as SDG. So things are going in and out in this loop. And each of those processes can have a different impact on CO2. For example, if we have lots of nitrogen stimulating primary production, the ultimate effect would be that primary production is removing the CO2, even if that looks like groundwater is a CO2 source. So I have been trying to test this hypothesis that groundwater is impacting the carbon cycle in those ecosystems, and I'm going to share some data from those 6 coral reefs, Indonesia, Australia, and the Cook Islands. So again, we have those time series, similar to the previous one. We have diol cycles for oxygen in green, and we also have the cycles for CO2 in orange. That they are a combination of diol and tidal. So we are trying to separate the contribution of those different sources of CO2, the direct inputs of groundwater, but also the alkalinity that's going to be a buffer to CO2. It's also the nutrients that are going to stimulate CO2 uptake. So the initial impression, if you just look at the coral reef data, is that there is a positive correlation between our groundwater tracer, CO2, and radon. That correlation was significant in 5 out of the 6 sites that we have these detailed data sets. The exception was Lord Howe Islands, where there is no correlation. There is lots of scattering in the data. Sometimes the correlation has these different populations, day and night, like Heron Islands. Sometimes that's a direct, positive, stronger correlation, and that's when we have lots of groundwater. For example, places like Nusa Penida in Indonesia and Rarotonga, the Cook Islands, they get fresh groundwater inputs that are super high in CO2, and then you have a stronger signal when you make measurements in seawater. Now, one of the things that is interesting is that the intercept of that radon to CO2 relationship is always approaching, or nearly always, except for one site, it's approaching the atmospheric equilibrium. So that means that if you don't have any groundwater signal, the CO2 in the coral reef, the flux to the atmosphere would be approaching 0. So the coral reef would be nearly in equilibrium with the atmosphere. Now, we try to quantify how those different pathways, those biogeochemical components can impact the CO2 process, the CO2 source or sink behavior of coral reefs. So we quantified the SDG flux in those 6 locations. We have measured the CO2 in the water, usually a little higher than 400s, roughly the atmospheric equilibrium. And we also quantified the fluxes of those 4 things that can impact CO2. From the direct CO2 inputs, TA, of course, or alkalinity is going to decrease the CO2. DOC can be broken down into CO2, and nitrogen is going to stimulate production and therefore remove some of the CO2. So with all of those assumptions, the initial perception that groundwater is a clear source of CO2 kind of shifts. So the net CO2 flux, after accounting for those 4 biogeochemical components, can be anywhere from slightly positive in 3 locations to a big negative, because the nitrogen input is overwhelming, and that stimulates primary production beyond the location where CO2 penetrates those lagoons. So we can also try to do the analysis that we spoke much about that yesterday, the TA versus DIC plots. It's quite messy because there is a big natural variability in the concentrations of DIC and TA in those different types of samples, going from bores, groundwater bores, seeps that you can sample on the beach, and transects as you're kind of penetrating the ocean. The background on those plots here is pH isoline, so pH of 6, pH of 7, pH of 9. So all those plots, they have the different pH on the backgrounds. So sometimes it's a little obvious, sometimes it's a mess, but what we see is that usually our groundwater samples that plot here, they tend to cross those pH lines as they get in contact with the surface waters. So what we see from this analysis is that groundwater seepage would have an immediate impact on the pH of the receiving coral reef waters, and that will drop the pH. So that's reducing the buffering capacity and the pH of the receiving waters. But how big is that impact? We can also look at that slope, the DIC to TA slope versus the SGG rate. And each dot is one of our locations. So if the slope is somewhere here, we have more calcification dissolution. If the slope is lower, presumably we have more photosynthesis respiration driving the chemistry of that coral reef. So there is some scatter, of course, there is large uncertainties as we try to observe those natural processes, but there is a clear correlation that SGG tends to stimulate photosynthesis and respiration before it stimulates calcification dissolution. So SGG is going to push coral reefs more towards that organic than the inorganic biogeochemistry. But that impact doesn't go very far. So if you summarize all of that data, we have an initial impression that SGG is a CO2 source. So very close to the shoreline where SGG is going on, I would say within 100 meters in those 6 locations, we have a perception that SGG is a CO2 source. But then that nitrogen coming from groundwater is going to drive primary production, and therefore, at the end of the day, there is much more nitrogen proportionate to CO2 that we're going to drive CO2 uptake further away from the groundwater source. So those ecosystems are complex. The initial perception that groundwater is a clear CO2 source that actually shifts when you consider all those biogeochemical sources, including alkalinity, DOC, and nitrogen. So to wrap up, SGG, with our broad definition, is widespread in coral reefs. Sometimes we have fresh groundwater, depending on the geology. Sometimes we have only tidal pumping. But everywhere we found some radon that's coming from that contact with the sediments. So the sediments are releasing radon to coral reef waters, and that's our tracer for SGG. We have large uncertainties. We have observations from just a few locations. There is a very large natural variability. The impacts on CO2, ocean acidification can be either positive or negative, depending on the local conditions. On average, SGG, it does feel like an overlooked carbon pathway. It appears to be a source, but if you consider the nitrogen inputs, it's actually a sink, after all. So that's what I had to talk about. I'll stop here. I'm happy to engage in discussions about those complicated processes. Thanks, Nadine, for organizing this and inviting all of us to be here.